Hello everybody and welcome to this video where we're going to be looking at more about enzymes specifically. And specifically what we're going to be looking at is what affects the rate of an enzymatic reaction. So these 5 things right here are the things that influence how fast an enzyme can catalyze a reaction. So temperature, pH, the concentration of substrate, the presence of cofactors or coenzymes, or the presence of inhibitors. These are all things that can make an enzymatic reaction happen faster or happen slower. So we're going to look at each of these in a little bit more detail. So the first thing that can influence the rate of an enzymatic reaction is temperature. So let's look at this graph down here first, and then we'll talk about some of the text. So you can see, as I increase temperature, you can see that the enzymatic activity, or the rate of an enzymatic reaction, goes up and up to a certain point. Now this makes sense because you can imagine as things get hotter, that means the molecules, so more specifically the enzyme and the substrate, they're going to be moving around faster, right? And if molecules are moving around more quickly, there is a higher chance that the substrate collides with the active site of the enzyme. So the faster our molecule move, the more collisions we're going to get between the active site of the enzyme and the substrate. However, once we reach a certain point, you can kind of imagine that molecules are moving too quickly, and the speed at which molecules are vibrating in a protein is stronger than the hydrogen bonds and the ionic bonds that are keeping the protein folded. So therefore, at some point, the high temperature will actually denature the enzymes, and the enzyme protein will unfold. If the enzyme protein unfolds, it loses its shape, and therefore the protein will lose its structure, and therefore it won't be able to do the same job it could do before. Which means after a certain temperature, the rate of this reaction plummets because the enzyme does not have its shape anymore, and therefore it cannot do its function. So this kind of relates back to living things in the sense that, if you know, animals can either be cold-blooded or warm-blooded. So warm-blooded animals, which include humans, are creatures that we keep our body temperature constant using homeostatic mechanisms. And a lot of the reason we do this is because our enzymes in our body will only work best in a very narrow temperature range. So we want to be in that sweet spot where we're going as fast as possible, but our body temperature is not too high that it denatures our enzymes. Cold-blooded animals, so things like reptiles, their body temperature is dependent on the temperature of the environment. So they will often warm themselves by sitting in the sun or cool themselves by going in the shade to help maintain the perfect temperature for enzymatic reactions. So the second factor that can influence enzymatic reaction is pH. So many enzymes work best at a pH of 7. However, some enzymes work better at other pH values. So for example, there's an enzyme called pepsin that is found in the stomach, and therefore it works really well at acidic pHs, pH of around 2, which is the pH of the stomach. Another enzyme called arginase works better at a slightly basic pH, but most work well in this kind of narrow pH range around 7. So the reason really extreme acidic or really extreme basic environments aren't good for most enzymes is because you can imagine that the enzyme shape is influenced by the presence of H+ or OH-. So remember, H+ is associated with acids, OH- is associated with bases. And we already know that things like positively charged and negatively charged R groups attracting one another is something that helps proteins to fold. So therefore, if I'm throwing a lot of charges like H+ or OH- into the mix by being in an acidic or basic environment, this is going to interrupt the attractions that help proteins to remain folded, and therefore the protein will either denature or fold into a different shape if there's lots of extra ions around. So the next factor that can influence the rate of an enzymatic reaction is the concentration of the substrate, so how much substrate you have to work with. So it kind of makes sense that if you have more substrate, the reaction can go faster. And that makes sense, because if you have more substrate, there's more substrate around that could collide with the active site of the enzyme. So therefore, as you have more substrate, the reaction becomes more likely. However, at some point, as you increase substrate, the rate of reaction stops increasing and actually remains constant. And this point where it levels out is called the point of saturation. And this happens because at some point, all of the enzymes are already in use. So even if I add more substrate, all of the enzymes are already bound to substrate and breaking it down. So adding more substrate won't make the reaction happen faster because all of the enzymes are currently being used up at that point. So even if I keep increasing substrate concentration, the rate of reaction will not increase anymore, but instead stays level at this point of saturation. The next thing that can influence enzymatic rate is the presence of cofactors or coenzymes. So these are molecules, and they're often vitamins, that they basically help enzymes have the correct shape. So remember that proteins are very reliant. Their shape is incredibly important for their function. And you can kind of imagine if an enzyme's shape changed, it wouldn't have the correct active site and therefore wouldn't be able to perform its function. So certain enzymes require molecules that are called either cofactors or coenzymes to help them keep the right shape. These molecules help them fold properly. So in fact, a lot of the reasons that you have to eat the vitamins that you need is because many vitamins help your enzymes fold properly and help your enzymes to maintain the correct shape. Vitamins are an example of a coenzyme. So finally, the last thing that can influence enzymatic rate is the presence of inhibitors. Now, inhibitors are molecules that interfere with enzymatic activity. So in other words, if an inhibitor is present, it will slow down the rate of an enzymatic reaction. So I'm going to show you an example right here. So in this example, my purple molecule, this guy right here, is the enzyme, and this is the substrate that we're trying to break down. So normally this substrate will bind to the active site and the enzyme will be able to convert it to a different substance. However, if we have an inhibitor present, what happens is an inhibitor inhibits or stops the substrate from being able to bind to the enzyme. So this can happen in one of two ways. So first, we can have what's called a competitive inhibitor. And a competitive inhibitor will compete, hence the name, with the substrate for the active site in the enzyme. That's what we see right here. So you can see my inhibitor binds to the active site, which prevents the substrate from binding. The second type of inhibitor is a non-competitive inhibitor. And this is a molecule that binds the enzyme on a different spot and changes the enzyme shape so it no longer has the same active site. So you can see in this case, this little yellow molecule is a non-competitive inhibitor. It's binding the enzyme somewhere else, but you can see when it does that, the active site looks a little bit different than it did before, right? The way the protein folded changed a little bit when the inhibitor bound, and therefore the substrate no longer can fit in the active site, so the reaction does not happen. Now, a lot of poisons that result in death are often examples of inhibitors. So what happens, a lot of poisons act by acting as inhibitors and binding with enzymes so that way our enzymes can't do the job they're supposed to do in the body, which results in death. It's also important to note that we had a lot of inhibitors in our body that are intentional. Like sometimes you might want to turn off an enzyme because you don't need it. So I'll give you an example. So imagine that this enzyme broke down triglycerides into fatty acids and glycerol. Now, if we didn't want our triglycerides to be broken down, we would want this enzyme to be turned off. So therefore, our body might make an inhibitor to turn off that enzyme. And then when it wants to turn the enzyme back on, it can get rid of the inhibitor, allowing the enzyme to do its job. So sometimes inhibitors are unintentional, things like poisons. However, sometimes they are intentional, and the body makes them on purpose to help turn enzymes on and off when needed. So that's it for this video, and I'll see you in the next one.